Thanks for joining us, everyone. We'll be getting started momentarily. Good evening to each of you, and good afternoon for our Hawaii families. Thanks so much for joining us for AskMe, our educational webinar series. My name's Audra Hines, Vice President of Care Services for the Golden West chapter of the ALS Association. May is ALS Awareness Month, and we have so much going on at the chapter. We invite you to visit our Golden West chapter website to learn more facts about ALS and check out ways that you can take action to help defeat ALS. There are so many ways to get involved in supporting the ALS community in California and Hawaii. Please do join us. I want to express our very sincere thanks to all of our industry partners and our mission benefactor families, including the Ron Malone Fund, the Morris Family Caregiver Initiative, and the Feig family, to name a few. The Golden West chapter is able to host an impressive number of these webinars, as well as build upon our programming for the future because of the generosity of donors like these. In a recent article, it was reported that ALS is the most frequent adult onset motor neuron disease. When we pause for a moment and think about that, it helps better inform our understanding of the prevalence rate of the disease. As more individuals are diagnosed with ALS, it's critical for these families to understand where to look for resources to help with managing life after a diagnosis. Having access to the best multidisciplinary specialty teams, as well as ALS clinical trials, is at the forefront of obtaining quality care and improving life with this disease. The Golden West chapter is proud to partner with more than 30 ALS clinics and centers in California and Hawaii to ensure that everyone living with ALS is referred to medical experts who know how to manage and treat ALS. Many physicians who are ALS clinic directors and researchers, like the speakers you'll hear from tonight, are committed to providing the most up-to-date treatment options, including research study and clinical trial information for those who are interested and qualified. Whether you're a person with ALS, a family member, caregiver, or friend of someone living with the disease, we want to ensure that you're connected with an ALS clinic team that's knowledgeable about the disease. For more information, please reach out to your care manager or visit alsogoldenwest.org to get connected. Tonight, we invited some special guests to help guide and inform us on the latest and available ALS treatments and therapies, as well as some that are still in the research pipeline. You'll hear two presentations tonight, and we'll take your questions after both speakers have presented. First, I'd like to introduce Dr. Jeffrey Rosenfeld, who's a professor and associate chairman in the Department of Neurology and the director for Neuromuscular and ALS programs at Loma Linda University Health System. Dr. Rosenfeld leads the Certified ALS Treatment Center at Loma Linda, and he's also a member of the California ALS Research Network. The members of that network were honored with the Golden West chapter's SE Commitment to Cures Award earlier this year. Thanks so much for being here, Dr. Rosenfeld.
Well, it's my pleasure. Thank you very much, Audra, and welcome. Can you all hear me okay?
Yep, hear you just fine.
And great. Hopefully you can see my slides. I have to apologize for a moment in advance because I'm on my backup computer. For some reason, my usual Zoom computer is not Zooming tonight. So I appreciate your patience, as I hopefully will not have any technical glitches here. And it's really my pleasure to be here tonight. I appreciate the invitation. And I love the idea that the name of this webinar is Ask Me. I jokingly told some colleagues yesterday when I realized that there'd be an opportunity to present some slides I didn't realize it was tell you, thought it was ask me, so I thought it was just questions. But I'm going to start out by speaking for maybe about 15 or 20 minutes about some of the exciting developments and as well as some of the controversies in current treatments and therapies for ALS. And then really what I find the most interesting is to hear your questions. So I want to leave plenty of time for that. So we'll start out with talking about disease heterogeneity, because it not only is a personal interest of mine and better understanding the disease, but I also think it provides a really important window into better understanding the future directions for therapies. And we're going to talk a bit about the difference between disease-modifying therapy and symptomatic therapy, and then where these drugs came from. I don't think I have to tell you all, this is an incredibly exciting time for motor neuron disease and for ALS. And just by virtue of having now 4 or 5 different therapies that are approved for treatment of ALS is something that I have never been able to say in my career so far. So it's very exciting, and they're not all the same therapy, and I'd like to give you some perspective on that tonight. So just by way of a very brief overview, I realize I'm probably preaching to the converted here, but the diagnosis of ALS is very dependent upon seeing certain very specific things clinically, that we see problems with the upper or brain motor neuron, similar or different problems with the lower motor neuron, and that there's some progression. And this triad of things, the upper, lower, and progression, is really what we look for as clinicians to make the diagnosis. The truth is, though, that there's a lot of variability, and probably if we were in the same room instead of on Zoom, you could look around with this number of people and see many, many variations of what this disease looks like. It's my contention that they're not all the same. That is, that there might be different drivers, and that may actually be one of the stumbling blocks and challenges in understanding the best way for therapeutically advancing this. The upper motor neuron is the brain motor neuron. It sends the signal to the spine, which is the lower motor neuron. The brain tells the spine what to do, and the spine tells the muscle what to do. In the clinic, we look for very specific problems with either the upper and or the lower to make the diagnosis. But the reality is, and this is a slide, a publication that I've borrowed, that shows there's a lot of variability. Sometimes we see mostly or only upper motor neuron with very little other than some electrical changes lower. And sometimes we see lower motor neurons, sometimes just bulbar, and then sometimes generalized. And it's important to understand these differences, because there's nothing to say that all of these forms or variations or variability in the presentation will respond to the same medicines. This is a slide I borrowed from a recent presentation that Dr. Harms did. He's in Columbia, and it just shows the number of publications and interest in this area. And it shows that since 1980, there have been over 33,000 publications in the scientific literature for ALS. This is a huge upswing, and you can kind of see that the rate of publication has really escalated. And if you look at different points along this curve, there are 3 inflection points. So the first arrow at about 1993 is when the SOD1 mutation was identified. And this led to a tremendous interest in understanding what we thought at the time might be the cause of the disease alone. And then in about 2005, the TDP43 protein was identified as an important pathology that might drive ALS, and that led to a big, steep increase in the number of publications and research interest, and also therapeutic advances. And then the last, at about 2015, is where the C9orf72 mutation was identified, linking forms of frontal temporal dementia to certain types of motor neuron ALS presentations. So there were some hallmark discoveries that increased this publication, but it's an astounding and rapidly increasing publication. I can imagine if this graph is moved further to the right in subsequent years, the bars will even get taller. It's important to understand that there are some excellent, phenomenal hypotheses about why motor neurons die. Neuroinflammation, axonal transport deficits, oxidative stress, stress to the endoplasmic reticulum, mitochondrial dysfunction, glutamate, and they're not just ideas. There's a lot of experimental evidence that suggest that if I had time and this seminar was about any one of these, by the end of the seminar, by the end of the webinar, you would probably be convinced that that was the driving hypothesis. But there are multiple ones. So either there are multiple right answers, or this is very misleading. But there's an abundance of evidence that these different underlying pathologies might actually drive motor neuron death. Now, if there's more than one right answer, you might figure that there are multiple pathologies, and perhaps some of them are more relevant to a certain stage of the disease. Like, oxidative injury might set up glutamate toxicity, which might set up some stress to the endoplasmic reticulum and the mitochondria, so they may be interrelated. But what I like about this picture is it suggests that we can't really address therapeutically just one at a time. It's too likely that there's more than one driving hypothesis, and it's distinctly possible that different presentations of motor neuron disease are driven by different underlying pathologies. So choosing a drug or a therapy to treat your motor neuron disease, you have to be very mindful about what you're treating. And each one of these has a very significant experimental basis to believe it's accurate. So it's been an enigma to us as to whether any one of these is the most right, the most prevalent, the most biggest target to go after, or are they all of similar importance? And then this is a purposely busy slide to show you that some of these pathways of pathophysiology, the same mechanisms I just showed you, are interrelated. One can trigger the other. If you'll notice, look closely, there are some stars printed on this cartoon of a cell. Each of these stars highlights a different mechanism of one of the available drugs that we have. So the one at the top that says glutamate next to it, that's the action of riluzole. And the one in the middle that says oxidative stress next to it, that's the action of what we think the action of radicava is. And the one that says mitochondria up on the left side, that's what we think the action of Relivrio is. So the takeaway message is we currently have 3 disease-modifying therapies, and they're not the same. They actually affect the cellular metabolism and potentially 3 different pathologies in the cell at the same time. We don't know if all 3 are better than taking 2 that are better than taking 1, but we do know that in, for example, the Radicava clinical trials that led to the approval, all of the patients were taking riluzole. And in the Relivrio trial, they were allowing for riluzole and Radicava. So we kind of did that natural experiment, but it wasn't systematically evaluated. It makes sense to think that addressing more targets has a greater chance of preserving the health of the motor neuron. And this highlights that by showing that there's multiple different mechanisms. I won't spend much time on this, but there are many, many genetic mutations that have been identified, and each mutation seems to be relevant to one of those mechanisms. The letters that are underneath in the boxes, those are the individual mutation or the genes that are implicated. But it's fair to say that the field got a lot more complicated than when I started, because now we have multiple mechanisms. When I got started, there was just maybe 1 or 2, and now there are an abundance of genetic abnormalities, each one relevant to at least one mechanism. And so we don't know if these genetic abnormalities will drive the disease or perhaps just in selected patients be the predominant pathology. But it's a bit more complicated, and it certainly highlights the need for potentially more than one therapeutic drug at a time to see if we can address more than one pathology at a time. So this slide is to highlight for you the differences between the phases of experiments, because I think tonight, depending upon how the conversation goes, we'll refer to some things as a phase 1, 2, and 3. And it has to do with the development of a new medication. When a medication is first introduced in the clinic, it's usually run in a phase 1 trial. And all we're doing in a phase 1 trial is using healthy volunteers to see if it makes anybody sick. Is it safe? Is there an obvious reason not to go forward? If it passes phase 1, phase 2 is often done with patients, and it looks at both safety and a little bit of whether the drug is working, although the purpose is not to see if the drug is working. The purpose is to see if it's safe. And phase 3 is actually to see if a drug works. And it's based on phase 3 that a drug often gets approved or denied. Recently, we've seen some approvals that came out of phase 2, but that's been a very unusual and recent development. And we can discuss that if there's interest. But I wanted to highlight these phases of the trial because I think it's relevant to what you're reading about. I also want to mention to you that therapy and treatment are often not the same. And certainly, I'm sure I'm preaching to the choir here, but multidisciplinary care is really critical. It should be viewed as critical to receive a multidisciplinary approach as finding the right medication. What we know in the data, the available data, are something as simple as a BiPAP machine or as straightforward as a PEG tube can actually impact survival more than some of the medications that we have available and some of the medications that we looked for and tested and didn't make the criteria. So I just want to highlight that it is a very important treatment modality to consider proactive multidisciplinary care. I'm going to mention, while I'm thinking about multidisciplinary care, the drugs that we have available that I'm sure we're going to be discussing in the question and answer are what we call disease-modifying therapies, which means it's been shown that the disease will change the course of the drug will change the course of the disease. Patients will decline less than they might have been expected to without the drug. That is not the same thing as a symptomatic medication, meaning the drug will make you feel better. So the drugs that we have for the most part available might not make you feel better, but they might make you do better. As in, your progression might not be the same as it would be otherwise, might be lessened, but it doesn't necessarily turn the clock back. So what I try to highlight in just a few minutes of comments is the importance of combination therapy, because we have multiple hypotheses, multiple reasons that motor neurons die, and it's complicated by multiple mutations that might drive those pathologies. There's a huge swing that has not been the case for very long in personalized approaches. That is, understanding better what particular drivers might be behind a specific patient's condition, and then finding the medications for that. And then, as mentioned here, non-pharmacologic care, multidisciplinary care, emotional mental health, and so on. And I would put a plug in for future disease stratification, which it's difficult for me to talk about in such a short talk, but understanding that all ALS might not be the same could in fact be a very important venue for understanding new drug development. Perhaps drugs that we passed on earlier might have had a selective benefit in a particular subset of ALS patients, and certainly drugs for the future might also as well. So I'd like to hold here. I think the plan is to either take questions now or have Dr. Goyal make some comments relevant to her stem cell trial.
Yeah, that's great. Thank you so much, Dr. Rosenfeld. We are going to take questions at the end of her presentation.
Oh, okay, fine.
And I'll just ask them out loud for both of you, so that's perfect. So yeah, this is a great opportunity. I'd love to introduce Dr. Namita Goyal, who is a board-certified fellowship-trained neurologist at UC Irvine, and she specializes in the diagnosis and treatment of neuromuscular disorders. She also serves as co-director of the UCI Health ALS Neuromuscular Center, and they are one of the clinical trial sites for a medication called Neurone that's currently being studied for the treatment of ALS. We're thrilled you could join us tonight too, Dr. Goyal. Thanks so much for giving this update.
I'm very happy to be here. Thank you to the ALS Association Colton West chapter for hosting this great webinar. I learn from Dr. Rosenfeld all the time, so I'm happy to be here with him. I was asked to speak about the Neurone Brainstorm study, and I'm happy to share these results with you. This is data from their phase 3 study. And as many of you know, the premise of Neurone is that they take stem cells from a patient's own bone marrow. These stem cells are then taken to a lab where they are isolated, they're expanded, and they're treated with Neurone. Now, the treated stem cells are injected back into the spinal fluid via a spinal tap or lumbar puncture. And I think of these treated stem cells as a vehicle essentially delivering Neurone or these treated stem cells to treat the motor neurons that lay in the brain and the spinal cord, hopefully helping their environment to create a better, healthy environment for motor neuron survival. Neurone technology has been under clinical development for 10 plus years. And you can see here that the phase 3 study was completed in 2020 with the results published in Muscle and Nerve. Now, this is the design of the phase 3 study. And what's important to note is that patients were evaluated for a 20-week pre-treatment period before the treatment actually started. And this becomes important as we're looking at the data, and we'll talk about this more. But for the first 12 weeks, patients were evaluated to see if they declined on their ALS functional rating score. If they had a 3-point or greater decline, they were deemed eligible for the study, and they were randomized to receive either Neurone or placebo into a one-to-one fashion. All patients had a bone marrow aspiration where their stem cells were retrieved, and over an 8-week period, these stem cells were treated with Neurone and then were ready for injection back into the patient. So patients either got Neurone or placebo. They received 3 treatments in every 2 months. So at week 0, at week 8, at week 16, and they were followed to the end of the study until week 28. Now, patients were eligible for the study if they had a score of 25 or greater on the ALS functional rating scale. And as you know, this scale has 12 questions. Each question is graded from 4 being full function to 0 being no function. And so a patient with absolutely full function could have a total score of 48. Well, in the study, in that 12-week pre-treatment period, patients had to decline by at least 3 points or even more in order to qualify for the study. Other important factors was they had to have symptoms for less than 2 years and a breathing test vital capacity of greater than 65%. Now, the study looked at a number of endpoints or outcome measures, including the rate of decline in their ALS-FRSR. They looked at the change from baseline in their ALS-FRSR. They looked at survival, vital capacity, and a number of biomarkers in the blood and the spinal fluid. Also important to note that there was a pre-specified subgroup analysis on patients with less advanced ALS. So those patients who at the start of treatment had an ALS-FRSR score of 35 or greater, they wanted to see how these patients did also. Unfortunately, the study did not meet the primary or secondary endpoints, but in that pre-specified subgroup, so those patients with less advanced ALS, there was actually a clinically meaningful treatment response seen, which I'll show you in some upcoming slides. Importantly, the analysis has started to suggest that the primary and secondary endpoints may have been missed because there were a number of patients with advanced ALS even at the starting of the study, because patients progressed in that 20-week pre-treatment period, there were a number of patients that actually scored a 0 in some components of the ALS-FRSR questionnaire, making it difficult to evaluate for further progression in that scale. So if someone hit 0, it was difficult to analyze their data and see if they further progressed. making it a floor effect, which we'll talk about in some upcoming slides. Importantly, NurOwn had favorable safety profile. And also very important to note is that when they looked at the spinal fluid in patients that were treated with NurOwn, patients had less inflammation, they had a reduction in neurofilament levels, and they had an increase in a number of growth factors that are thought to protect the motor neurons and create a healthier environment for survival. So let's now look at two important endpoints, the rate of decline in the ALS-FRSR, as well as the change from baseline. And these endpoints are important because these are the endpoints that the FDA is also interested in. So when we talk about the rate of decline of a patient, what we're talking about is that pre-treatment period. If you recall, I said patients were monitored for 20 weeks before they even started treatment. And during this period, this is an example of a patient in the study. You can see that this patient declined here by 1.43 points per month. And you can see the decline in their slope. However, post-treatment, you see that their slope actually improves to a measure of 0.09. Well, if you take the post-treatment and subtract it from the pre-treatment, you can see here that you get a value of 1.52. Any patient in the study that had an improvement in the slope, like you see here, of greater than 1.25 points per month was deemed a responder. So we'll talk about the responders. Now, you can see here, when you look at all participants, you actually, in blue are the NurOwn treated patients and in red are the placebo group. There is not a significant difference in the responders in all participants. However, on the right side, you see the patients that had less advanced ALS, so a score of 35 or greater at the starting of treatment. Well, you're starting to see almost a 20% greater responder effect in the patients that received NurOwn in comparison to placebo. Now, similarly, when you look at the ALS-FRSR score, so from baseline, the start of treatment to the end of the study, what you see here is that patients that were less advanced had a 2-point preservation or greater in comparison to the placebo group. So patients that were treated with NurOwn progressed less by 2 points or more in comparison to placebo. This was not seen in all participants, and we'll discuss why. But importantly, when you look at the patients with less advanced ALS, you see this treatment effect across all time points starting week 2 through week 28, where you're seeing a 15 to 20% responder effect favoring NurOwn. And additionally, again, in this ALS population of less advanced ALS, you also see a 2-point preservation in their decline from their ALS-FRSR score in comparison to the placebo patients. And this is seen from week 12 through week 28. Some important data that is noted. So why aren't we seeing this across all patients? Well, unfortunately, in the study, they did enroll more advanced ALS patients than what we typically see in clinical trials. In fact, on the left, you see a number of clinical trials and the average ALS-FRSR score of these patients. In red, you see the therapies that have received FDA approval. And you can see that their average score ranged anywhere from as high as 42 to the Amylyx score of 36. Now, unfortunately, in the NurOwn study, they enrolled patients with a much lower score with an average score of 31. And when they looked at all patients, they realized that almost 25% of patients had a baseline score, so at the start of treatment, of less than 25. Now, what's important to note is that when patients hit a score of less than 25, in this case, in the NEURON study, approximately 40% of patients had at least one value that was scored at 0 in these subscales of the ALS-FRSR. Now, as you know, once you hit 0 on the ALS-FRSR, the scale won't detect further progression. And so essentially, because the scale, not necessarily the treatment, but the scale couldn't detect further progression, it's essentially a potential misclassification of how patients responded when they had a score that was lower than 25. So another example of this floor effect is what it's called, is if you look at a patient from the PROACT database and from the NEURON study, if patients scored a 0 on at least 5 or 6 items, you can see here before their ALS slope is declining, but once they hit 0 on 5 or 6 items, it almost looks like their score has plateaued or hit what we call a floor effect. And unfortunately, we know that patients do clinically progress, but the ALS-FRSR scale isn't able to capture that progression because of this floor effect. So what's also important to note is that the pre-specified data looked at patients that were 35 and greater, so less advanced ALS, but when they looked at patients with a score of at least 26, even through 35, they do see a responder effect by 15 to 20% greater in the NEURON-treated group from placebo. And similarly, when they look at patients, again, with an ALS-FRSR score starting off from 27 through 35, there's at least a 2-point preservation in the decline in their ALS-FRSR from the starting of treatment to the end of treatment, indicating that NEURON may be effective in this population as well. So as long as they have a score of greater than 25, we are seeing a treatment effect. Important to note, I'm going to draw your attention to the right side of the PowerPoint presentation, where you can see in the study, if someone had a score of 25 or less, 100% of the patients had at least one item that scored 0 on the ALS-FRSR scale, again, making it difficult in those patients to evaluate for true progression. So let's eliminate the floor effect. Well, let's look at the patients that had no items that scored 0 at the starting of treatment. And what you actually see here on the left is that almost a 20% difference is seen in a responder effect greater for the NEURON-treated patients than placebo. And similarly, when you remove the floor effect, so taking only the patients that had no zeros at the start of the study in the ALS-FRSR score, they have a 2-point or greater preservation, so less of a decline when they're treated with NEURON. And this was 106 patients in the study. So what I want to also draw your attention to is when you look at patients with less advanced ALS, so if they started off with a score of greater than 35, or if you took all the patients, as long as they didn't have any item that scored 0, so no evidence of floor, you actually see similar effects. On the top graphs, you see almost a 20% responder effect favoring NEURON. And in the bottom graphs, you see in both groups, there's a 2-point preservation with less progression in the NEURON-treated groups. And lastly, when you look at their spinal fluid, you actually see on the top left that across time points, these patients that were treated with NEURON in blue had less inflammation. They had a decrease in the neurofilament levels, suggesting less neurodegeneration. And in the bottom graphs, an increase in neuroprotective markers, indicating that neuron is meeting target engagement and doing what it's postulated to do in benefiting potentially the motor neurons with less inflammation and more neuroprotection. The study, in fact, was the largest biomarker spinal fluid study conducted in ALS. There were 45 spinal fluid biomarkers collected and at 7 different time points. So each patient had 7 spinal taps throughout the study. So in summary, I hope I've shown you how the primary and secondary endpoints were actually confounded by the advanced ALS patients scoring a 0 and essentially having a floor effect where it was difficult to measure disease progression if they started off with a 0 at the beginning of treatment. Now, if you took the patients without a score of 0 at the starting of treatment, or the patients with less advanced ALS, we are seeing a treatment effect with benefit of neuron and less ALS progression in these patients. And importantly, when we looked at the spinal fluid, we saw an increase in markers of neuroprotection and a decrease in markers of neuroinflammation and degeneration compared to the placebo group in all patients that were treated with neuron, irrespective of their ALS-FRSR score. This study was conducted at 6 centers throughout the US listed here, and including 3 centers here in California, UC Irvine, Cedars-Sinai, and CPMC. Interestingly, the FDA has decided to do an advisory committee meeting where they are going to review the license application for neuron, and this is postulated to happen in the upcoming months. And so I want to thank you all for your attention. I want to thank the patients and their family members who participated in the study and spent countless hours in the study, and the Golden West chapter for hosting this webinar. And I'm happy to answer questions. I'll stop sharing.
That's terrific. Thank you so much, Dr. Goyal, and to Dr. Rosenfeld. Terrific presentations by both of you. We do have a few questions in the Q&A. I'll ask these out loud so that everyone can hear them. And for those in the audience who still have a question, go ahead and type it into the Q&A function at the bottom, and we will get to your question as soon as we can. Piggybacking off of what you were just discussing, Dr. Goyal, someone's asking, do these results indicate that starting treatment immediately upon ALS diagnosis is important?
Yeah, I think that's a great question. We initially saw that in the patients, especially in that group with an ALS-FRSR score of greater than 35, so less advanced ALS, we are seeing that treatment effect. But what we learned after the study was completed and after we analyzed the data is that we're actually seeing a treatment effect even in those patients with a score of greater than 25. So these are moderately affected patients. And the only reason we believe that we may not be seeing a treatment effect in the patients with even more severe ALS is not because there may not be a treatment effect, but because there is a flaw in the ALS functional rating scale and able to capture or measure disease progression or that treatment effect. So we are actually seeing a treatment effect in patients with even moderately affected ALS. And then what I'll also add is what's important to note is the spinal fluid data is showing us that there was an improvement in inflammation, meaning a decrease in inflammation in all patients that were treated with neuron, even the more severe ALS patients.
Great.
If I could just add a comment to that, it is a good question, and it highlights the kind of, in a bigger picture, not specific to this trial, what we're trying to do by introducing stem cells is really to change the neighborhood, to change the environment of the surviving motor neurons, to make it a more hospitable place. And by keeping the surviving motor neurons alive longer, you enable them to compensate some for the neurons that are dying in the disease. So it makes a lot of sense, as you just highlighted, to treat as early as possible. I use the analogy, it's kind of like when you choose to put fertilizer on your lawn. It's better if it's green when you start, if you fertilize a brown lawn, you don't generally get green, you get more brown. But it's an idea of preserving the health of the motor cells. One of the misperceptions, I think, about stem cell research is that we are replacing the sick motor cell as if it's going to regrow back down to the muscle and function like the one that died. And that would be great, but there's very little evidence that that happens. It's much more evidence that they're neuroprotective and they're going to preserve the health of the motor neurons that have not yet died.
I see. And related to that, someone's asking if there's been any research with cord blood stem cells.
You mean cord blood, C-O-R-D, or core blood? I don't know what core
She says
cells
core,
are.
I think she might mean cord.
Yeah. Do you want to take, and Namita, do you have anything you want to say about that?
Not to my knowledge, you're talking about umbilical cord?
Yeah, I think that's what they're referring to.
Yeah. Not to my knowledge. I mean, the research that's being done so far is with mesenchymal stem cells. So from a patient's bone marrow, from an embryo, they can take stem cells, like a 6-week embryo, or adipose fat cells. And there's been some research with that.
Right. I would echo that there has been some interest on the internet about banking cord blood to create stem cells. And in some applications, it's been used, and maybe in non-regulated trials, that is not clinical trials, but independent clinics that pop up around, that has been tried. I have seen that before, but there are no data that we can report reliably about whether that's a good approach.
Gotcha. Thank you. Thanks to both of you. Someone's asking if there was a difference in neurone treatment for male versus female patients, and asking what the ingredients or components of neurone are, if you are able to share that.
Those are great questions. I have not seen any data in comparison of males to females. And then as far as neurone, it's a proprietary brainstorm treatment. And so I don't know the exact secret sauce, but what we are seeing in their spinal fluid is that there are a number of neuroprotective growth factors that are being analyzed. And so we do know that the cells are essentially treated with these neuroprotective markers to help, like Dr. Rosenfeld said, motor neuron survival of the healthy motor neurons.
That's great. Separate question, what kind of test is given to relatives of ALS to find possible ALS genetic markers? What are the markers? And is there a percentage known of relatives with ALS who have known markers contracting ALS?
I can try to take that question. So right now, there is no established test to give to a family member to determine if they have risks similar to their affected family member. However, what we do with family members is sometimes if we find a genetic marker, so to speak, or a genetic mutation in the patient, and the family is anxious, or the people that share DNA with that affected patient are anxious that they too might carry that marker, after some discussion about the pros and cons of the test, we are able to test somebody. What we're not able to do is to tell that person, if they have the marker, that they will definitely get the disease. We're not able to do that. There's a higher likelihood, if you have the marker, that you will get the disease. But I have cared for a family that the mother, who obviously had the marker, had several children, and they inherited that marker, and they actually died of the disease. And she was in her 70s at the time I last checked in with her, and she did not have apparent symptoms. So it's not a given. So to answer your question directly, I would say no, we don't have a blood test automatically for family. But when we do find a genetic mutation in a patient, we can look for it. in people that share that DNA.
That's great. And to piggyback on that for either one of you, separate questions, since ALS appears to be multiple pathologies, do you now believe that ALS involves more than one genetic mutation like C9orf?
Yeah, that's a very relevant question in this time. And I would say that, you're a little bit preaching to the converted, because I'm one that goes for cocktail therapy, and I'm one that promotes that we should be paying attention to these differences. But there's clearly indicators that there are gene-gene interactions within ALS. It's puzzling as to why a person would develop multiple mutations in relevant genes, but it's not so puzzling as to say that if you have a certain genetic background, a specific mutation may behave differently than if you had a different genetic background. So I do believe there are multiple mechanisms involved, and perhaps in the same patient. I think it's possible that there are different genetic drivers of those multiple mutations, but it's a little premature to say that everything is relevant to everything. I can say that right now, when we send a genetic test out on a patient, we're testing a panel of genes. It could be, I don't know how many are in the current panel, 30, 50, something like 30 or so, I think. How many is it, Namita?
30.
30,
Yeah.
okay, if I remember correctly. So about 30 different mutations. These are the ones that are called at-risk or causative gene mutations. And it's the same 30 genes. Generally speaking, we don't find multiple ones that are wrong in the same patient.
Mhm.
We don't. It can happen, but it's not common.
And adding to that, I think it's also important to note that if you have a genetic mutation, or if mom or dad had a genetic mutation and presented at an age of 75, what's interesting is that we are seeing that the same genetic mutation within the family can present at different ages. And, one family member may present at 40 and another may present at 70. And sort of piggybacking on that comment is, could there be other drivers that trigger a mutation to become symptomatic at a certain age? And is it some of these mechanisms that Dr. Rosenfeld talked about?
Gotcha.
I want to say one more thing about genetics in ALS. Up until recently, and this is probably not something I should be bragging about, but up until recently, I didn't really check genetics that frequently, except when it was relevant, if somebody said they had family members. But I've been very impressed with really a lot of data coming out that it's not that uncommon to find mutations in patients that do not have it run in the family. And I may have been a little bit slow to drink the Kool-Aid, but I will tell you that I now check every one of my patients that's willing for their genetic background. And among the most exciting presentations and advances that I have seen this year has been a recent presentation that I saw from the group in Colombia in a project called Silence ALS. And it was very impactful for me because I'll spare you the details, but they basically took a specific patient's mutation and developed a genetic therapy for that particular mutation. And they dramatically changed the course of the disease. So had they not known that there was a mutation, they wouldn't have known that there was a targeted therapy. But that specific personalized therapy might be something that we're going to be seeing a lot more of in the future. And that's why I want to know which of my patients have which mutation, and I'm screening everybody.
Yeah, and I think it's important to note for the patients that ALS genetic testing has become free. So you should make sure to ask your provider. We are recommending that every patient, irrespective of a family history or, depending on age, doesn't matter. We want everyone tested. And as you know, there's a new therapy for SOD1 that's been FDA approved. So it is important to have genetic testing and will help guide your treatment.
Yeah.
That was really helpful from both of you. Thank you. Shifting gears a little bit, someone's asking if there are current therapies or research in the pipeline that address PLS. She says those of us in that category are usually excluded from motor neuron disease therapies.
Right. Yes, I agree exactly. And I'm not aware of a specific targeted gene therapeutic trial, a drug trial for PLS. However, I know that from my own experience, I'm actually working with a small biotech company now that is going to be running some exploratory trials. And I have proposed, and others perhaps as well, but I certainly have proposed that we study an upper motor neuron or PLS group to see if there's a signal there. And as we better understand the heterogeneity of the disease that I tried to highlight in my comments, I think this is going to make more sense when we stop thinking of ALS as one thing and we say there's upper motor neuron disease, there's mixed, and there's lower motor, and there's bulbar disease. Perhaps we'll see more trials. So I can't tell you there's one currently, but I can say there's active discussions very currently to address the needs of the PLS population.
And yeah, I agree with that. And I think it's important to note that there was actually a natural history study going on with PLS, and it was multiple centers involved. And so whenever I see a natural history study, that actually excites me, because I know that companies and pharmaceutical companies are looking at this data to help try to find and develop a therapy, which is often why we do these natural history studies.
That's great information. Someone's asking with related to neurone, with the neurone's positive effects with participants with FRS greater than 25, will there be a follow-up trial to only focus on these higher functioning people? Assuming this shows the same success with this subgroup as the previous trial, would FDA approval likely be forthcoming, and how soon?
Yeah, no, that's a great question, and all questions that have been debated over this time period since the trial was completed in 2020. I think what I maybe didn't focus on was that, and what the trial design sort of flawed in was that they had an enrollment criteria of patients greater than 25 at the beginning or that pre-treatment period, but they didn't sort of have a bottom cap when the treatment actually started. So there was a 20-week period where patients potentially declined much, much more than 25 before even treatment was started. And so I think that's been some of the challenge in trying to understand the data and analyze the data. But now that we are seeing a treatment effect because of the ALS-FRSR scale issue in those patients with a score of greater than 25, they have actually submitted the data to the FDA, and the FDA has decided to do what's called an advisory committee meeting, which means that they aren't requiring another trial. They're actually going to review the data and see if the data is strong enough to get FDA approval. And this is called a biologics license application because it's stem cell, it's not a drug. But they are going for an application, and we'll hear more about it in upcoming months. It should be soon when the advisory committee meets.
That's helpful because we have a lot of questions sort of related to, you know, if I want to try neurone, how can I get it? Or when will that be available? How could I access that? So it sounds like we're waiting for that next committee meeting.
Yeah, it's a question that I almost get every single day since we started this study, and I wish I could get it for patients, but right now we are just waiting for the FDA committee meeting to know, you know, to understand the data even better, to give it a full review, to really have experts from across the nation meet and analyze the data and see if they do believe it is worth approval.
Great, thank you, very helpful. Shifting gears again, someone's asking, saying a couple of years ago, the University of Tel Aviv showed that damaged cells might be able to recover. What's the current status of that research?
So I'm assuming they're talking about some of the stem cell data, because some of the stem cell data and even the company Neuron, the company's name is Brainstorm, they are originated in Israel. So some of this data that this patient may be talking about is because years ago, the data was coming from Israel. And when I mentioned in my talk earlier that Neuron has been under clinical development for 10 plus years, the earliest phase study was actually started in Israel, and then the phase 2 study was done here in the US. So it's just further data indicating that maybe we are seeing motor neuron survival. Like Dr. Rosenfeld mentioned, the stem cells are trying to create this healthier environment so that other motor neurons that are not affected in these ALS patients can not be affected and patients may not progress.
That's great. Quick follow-up, Dr. Rosenfeld, for you. You mentioned silent ALS. This person's wondering if that's an example of what precision ALS therapy is and how can people access things like that?
Right. So it's very much a work in progress. This is not an established program that you can dial into. Precision approaches or precision medicine means a lot of things. In this instance, it did mean developing a specific genetic therapy for a specific genetic mutation that was relevant to that person. That's like the ultimate in precision. As we get further with some very active research, we're hoping that there are markers that would indicate which therapeutic way to go. I was involved in a project from a small company in San Francisco where we're looking at actually a new bioassay where we can take a patient's serum, for example, from their blood and run it on this assay in the lab, and it shows us a profile, and that profile perhaps would distinguish which therapy they might respond to. It's a little bit early in terms of knowing the results from that, but that's another non-genetic application of precision medicine. So the term precision medicine just means not everybody gets the same thing. It's specific, much like, if you have an infection, you get an antibiotic that supposedly responds to that infection. And that's kind of like a precision medicine. In the case of ALS, we're thinking it's going to be even more precise with that. So the question about how to get involved in that, I really don't know. I know that the Columbia program is a large program, and I don't know if you had a specific mutation, if they would or wouldn't be interested in it. I'm not sure. I know that I don't want to answer incorrectly. I'm just not sure if that's open to the general public or not.
That's fair. Yeah, and with all the currently available therapies, this person's wondering, the 3 R's, riluzole, Radicava, Relyvrio, what's your opinion of when to use which one first, and when do you add the second or third combination, if at all?
Yeah, well, the short answer is we're trying to figure that out, okay? And we're not really sure about that. I can tell you what we do practically, but this is not medical or scientific. This is just practical. Riluzole is very available. It's at the pharmacy. It's on the corner. If you write the prescription today, you'll have the drug tonight. So that's simple, okay? Radicava and Relyvrio come from specialty pharmacies, so they get shipped. And we don't know what the second medication should be. Generally speaking, at least in my hands, riluzole is the first medication because the patient can start right away. I give a patient sort of a loose background about the different approaches. And if they are inclined to want to take all 3, I would start them, but not all at the same time. I would start the riluzole perhaps for a week or 2 weeks, more likely, and then introduce the second and then introduce the third. But I have no order, basically. They're generally well tolerated. The comments I get back from when there are any adverse comments are different between the 2. So sometimes somebody tolerates one better than the other, and we go with that one. But there's no science here about which one to do first or which one to do first in a specific patient situation.
Yeah, I agree with that, and I probably follow a very similar practice practically. It's about which one can we get first, and we have a lot of data on riluzole, and it's easily available. It was FDA approved since 1994, so that's certainly one of the first ones I prescribe. And then for Radicava and Relyvrio, it's about which one will get prior authorization first. And then I advise patients that you might want to wait at least about a month in between starting medications just to monitor for side effects. It has nothing to do with a scientific starting one versus the other, but it's more about monitoring for side effects. So if there is a side effect, we would know which one to stop.
Sure. Question, someone says they're going on the Healy trial at the end of the month and it will go past their 2-year mark of having ALS. He's wondering if he would be able to take Neuron if it's approved after his, participation in the trial, if that's even a possibility.
So it's really the FDA that's going to decide the type of ALS patient that would qualify for Neuron. I mean, what we've seen with Radicava, Relyvrio, and riluzole is the FDA approved it for all ALS patients. What we've noticed practically is that with Radicava and Relyvrio, it's the insurances or the payers that sometimes put these stipulations on you have to have a certain criteria in order to get insurance approval. However, from an FDA perspective and a doctor prescription perspective, we often will just prescribe as long as the FDA approves it. So it's really going to be the FDA's decision on if they approve it. Do they approve it for all ALS patients or do they approve it for a certain subset of patients? But I think we'll learn more after the advisory committee meeting.
That makes a lot of sense.
And so I'm sorry, to answer the patient about Healy, typically the patient, it doesn't matter on symptom duration of a patient if the FDA approves it for all ALS. However, if the FDA says it's not symptom duration we're looking at, but you have to have a certain ALS FRSR score, these are all things that are to be determined.
Makes sense. Someone's asking if there's any consideration for direct application of Neuron through the brain instead of through the spinal fluid.
That's a great question because there are other stem cell trials that are injecting stem cells directly into the brain or directly into the spinal cord. Neuron has, in their clinical trials, never done that. In their phase 2 study, there was a point where they injected into the muscle and into the spinal fluid. However, in the phase 3 study, they decided to pursue just the directing directly into the spinal fluid. Now, what's important to note is that if you inject directly into the brain or the spinal cord, in those clinical trials, patients had to be immune suppressed. So they had to be given an immune suppressive therapy regimen. Sometimes that comes with a panel of side effects. What was nice about the Neuron study is that no immunosuppression was required. So you could just have the injection, and the study did show a very nice safety profile.
That's great. Thanks, Dr. Goyal. I have, I know we're wrapping up. I have an interesting question here. How do you know how many neurons are still alive?
Well, it is an interesting question, and there are ways to know. You remember when I showed you there are upper motor neurons and lower motor neurons. So the lower motor neurons are much easier to quantitate than the upper motor neurons because the extent to which those motor nerve cells are alive, they send their projection or their wire, axon we call it, down to the muscle, and you can actually count them. We don't do it regularly. It's very tedious and it's an electrophysiologic technique to do it, but there's a way. What we know, though, is it's proportional. Eventually, it's proportional to the degree of weakness and atrophy that a person has. So it's, assuming, let's just say you have 100,000 to start that go to a muscle, you might not notice the loss of the first 50,000 because the remaining 50,000 can compensate. But once you cross a certain line, perhaps it's much less than 50,000, you notice the loss more regularly. So we don't ever quantitate how many, we just get a qualitative sense that there's a lot more, a little more, or the same as when we started. So I'm sorry that's an indirect answer, but there is.
Yeah, no, that's great. Last question I have, which is also pretty interesting, asking if there's any possibility of using the samples submitted to 23andMe genetic testing for the presence of certain ALS markers. I know right now it doesn't test for that, but to possibly look at preventative measures.
Theoretically, yes, these are tests done on DNA. And I don't know that much about 23andMe, although what I do know is that they have a panel of things that they look for, a series of things that are of interest in the general population. Their panel of things that they look for in terms of mutations, to my knowledge, does not include the ALS-related genes. But I don't know specifically enough. Namita, do you have any idea of what the panel on 23andMe is overlapping the genetic risk for
No,
ALS?
I don't. And in fact, I was going to suggest that we've actually seen some 23andMe like questionable data. And so now that ALS genetic testing's become free, I would tell patients that you should go the route of the specific ALS gene panel, just to make sure that, the data is correct and that there isn't any mislabeling of genetic information. I worry in some patients that it could lead to, sort of a diagnostic odyssey unnecessarily.
That's great. That's so helpful. We got through a lot of questions, and I want to thank both of you for being here. This was a wonderful presentation. Thank you so much for doing this tonight. And I want to thank all of our people in the audience too. Thank you for being here, for being a part of our community. Just a reminder that during ALS Awareness Month and every month, we offer lots of ways that you can help raise awareness and support for the ALS community. Please join us in celebration of Lou Gehrig Day as we honor this special day with a few events. And additionally, many of our other fall events are now open for registration. Please visit calendar.alsagoldenwest.org for more details and to register. As you know, philanthropy fuels everything we do, and we're grateful to committed donors who give generously throughout the year. To learn more about how you can support the valuable programs we offer, including programs like this one, please visit our website at give.alsagoldenwest.org, and please watch for that short survey on tonight's presentation so that we can plan for future programs. Thanks so much, and we'll see you again next time.